Hello everyone, my name is Miquel. I'm originally from Barcelona, Spain, and today I'm here to talk about machine learning and what machine learning can do for medical informatics and healthcare data analysis. First of all, let me congratulate you. You've made it this far. We are in Module 5 already, and I really hope you are enjoying the course. As you might know, from now on, there's going to be an important component of machine learning in this course. Please do not panic, please do not run away. Machine learning can be easy if you know what you're doing. So the facilitators for the machine learning part of the course are going to be Victor Olago and myself. Victor is originally from Kenya. He did his bachelor's in information technology and a master's in epidemiology. Currently, Victor is a data scientist at the National Cancer Registry in Johannesburg, South Africa. Victor has been doing machine learning and record linkage for a living for the last couple of years, so he really is a good fit. He really is an expert in the field. I really encourage to reach out to him if you have any questions. Myself, my name is Miquel Duran Frigola, and as you can see from the many items here, I've had a more sort of erratic career, and the only reason I'm listing all of these items here is because I want to convey the idea that it's never too late to learn machine learning. It's never too late to learn how to code. I'm actually an organic chemist by training, and I did my PhD in biomedicine. Currently, I'm the lead scientific officer at Ercilia Open Source Initiative. This is a very small not-for-profit charity based in the UK. What we are trying to do is to disseminate and implement data science pipelines in low and middle-income countries. Back to the course. As you know, we have divided this course in a basic module, in a basic course, and in an advanced course. Today we are in Module 5 of the basic course, and I'm just going to give you, as I said, a very general introduction to machine learning. In Module 6, the next module, I'm going to focus on how can we apply the basic machine learning concepts to the record linkage pipeline that you already know. In the advanced course, we will get a little bit more hands-on. In modules 1 and 2, we will do 2 case studies where we will apply supervised and unsupervised machine learning methods. You will learn today what do we mean by supervised and unsupervised machine learning methods to the record linkage pipeline. In module 3 and 4, we will explore and discuss how can we package and document our code so that it looks more professional and essentially brought to production, that is implemented within our institutions. So before we get started, a disclaimer. Remember that the overarching goal of this course is not machine learning. The overarching goal of this course is record linkage. So don't expect to become experts by the end of this course on machine learning. Hopefully you will be experts on record linkage, but not on machine learning. There's a lot of extra materials nonetheless. You will find in Moodle that we are providing a lot of extra materials. So if you are really curious about machine learning, we really encourage you to explore those materials. There's a lot of great didactic materials out there, and we have done a selection for you. So everybody's talking about machine learning these days, and there are claims essentially in every field of biology, biomedicine, healthcare, and technology, actually. If we focus on healthcare, which is our field, right, you will find articles, you will find scientific literature applying machine learning methods to the identification and diagnosis of diseases, also to the improvement of the drug discovery pipeline, to medical imaging, to personalized medicine, for instance, in the analysis of genomes, to optimizing how we deal with healthcare records, and even with for disease prediction and anticipation of epidemiological events. As you might have grasped from my biosketch before, my expertise really is in drug discovery, and I have to say that in the last 5 to 7 years, I have experienced an avalanche of machine learning methods in virtually every step of the drug discovery pipeline. Maybe you know it. Bringing a drug to the market, discovering a drug, it's incredibly costly and takes many years, anything from 12 to 15 years, and billions of American dollars. So, of course, there's a huge need for methods that are able to make this process more efficient and optimize it. And I think that there are good tools that are applying machine learning everywhere now in this pipeline, from the very early stages, for instance, identifying the optimal genes to target given a disease, to the latest stages, for instance, the monitoring of adverse drug reactions once the drugs have been approved and delivered in the market. If you are not into drug discovery, but you just follow the news, or you are into Netflix, you might know about this documentary called AlphaGo. This is an incredible story. DeepMind, which is a machine learning company now owned by Google, developed an algorithm called AlphaGo that was able to beat the world champion in this very complicated game called Go. And there's a nice documentary about it. Importantly, the same company, DeepMind, very recently released an algorithm called AlphaFold that is able to predict the 3D structure of proteins only based on the sequence on the amino acid sequence of the proteins. This is a major scientific achievement because once you know the 3D structure of a protein, then you are able to design, for instance, small molecules that will be able to interact with it. And those small molecules are essentially good drug candidates if this protein is your target. Immediately when a few weeks ago I saw that the AlphaFold code was released, I downloaded it and used my laptop computer to predict the structure of my protein of interest, which is currently a Plasmodium falciparum target called PF-ATP4. Plasmodium falciparum, as you know, is the pathogen that causes malaria. So it's really important to try and discover new targets against this pathogen that will be able to kill it more efficiently and overcome resistance. This is one of such targets. So now, thanks to AlphaFold, we have the 3D structure of this particular target, and hopefully we will be able to design new molecules that will be able to interact with it, and those will be good drug candidates, we hope. Another interesting example is this picture called the Portrait of Edmond Bellamy, which was completely generated by a computer back in 2018 and was sold in an auction house for, I think, $500,000. What's interesting here is that there's no artist behind, it's just a computer creating art, right? And the type of methods that are being used for creating this type of art are called generative models. And actually generative models have been also applied in the field of drug discovery to invent new molecules. And a major achievement in this regard is a drug completely generated by an AI that has entered clinical trials. This happened last year from this company called Exentia. So again, when at Ercilia we saw this kind of algorithms coming up, we immediately applied them to the generation of new candidates against the Plasmodium falciparum parasite, the causative agent of malaria. And here you can see some examples. As you can see, the algorithm, the computer, is predicting that there will be a scaffold that will confer activity to small molecules, and then is generating alternatives in the branches of the molecule, right? If you have the eye of a chemist, you will recognize that these molecules are quite interesting and essentially good starting points for drug candidates. The bluer the background, the more active those molecules are predicted to be, so meaning that the computer is getting better and better at suggesting candidates. Okay, I hope that all of this sounds interesting, but of course the question, especially the question if you don't know how to code, and if you are not an expert in machine learning, is how does it work? How can I do that in my daily life? So for this, I think that the only concepts that you need really are these three keywords: supervised machine learning, unsupervised machine learning, and reinforcement learning. Supervised machine learning and unsupervised machine learning is what we're going to be using in this course because it's what's relevant for record linkage. If you know how to allocate your problem into a supervised or unsupervised machine learning framework, then for sure you will be able to identify the particular algorithm that is going to help you solve this problem. When we talk about supervised machine learning, basically what we have is labeled data. By label data means label for which we know the outcome, a training set, so to say. The two families of supervised machine learning are classification, yes or no, active, inactive, etc., or regression. When we talk about unsupervised learning, we don't have labeled data, we have unlabeled data. And what we are trying to do is we are trying to organize the data, for instance, by discovering clusters of samples that share some characteristics, or by doing projections. When we have very complex data, multidimensional data, we might want to project it in 2 dimensions so that we can visualize it better and sort of understand what is the underlying structure of the data. When we talk about reinforcement learning, I think that the image that should come to mind is a robot. So reinforcement learning is an artificial intelligence that interacts with an environment. The more it interacts, the more feedback it gets, and therefore the better it gets at the particular task. I think that this is best understood by examples, so let's go into some real-life examples and try to classify them into supervised, unsupervised, or reinforcement learning. First example will be labeling emails as a spam or not spam. I'm sure you've seen this in your email inbox. Some of the emails are labeled as potentially spam. In this case, there's an artificial intelligence behind, there's a machine learning method behind that is making essentially this prediction on the new sample, that is the new email that is reaching your inbox. Those methods have been trained on previous data, on labeled data, and therefore this is a case of supervised machine learning, in my opinion. Another example would be grouping scientific publications by topic, that is organizing the scientific literature by topic, by authors, by citations between papers, etc., etc., etc. Finding this kind of structure, it's a clear case of unsupervised machine learning. Classifying microscopy images as tumor or healthy, this is something that happens in clinical practice. Usually the way it works, a set of experts, a set of clinicians or physicians label existing images as tumor or healthy, and then those images are fed to a machine learning algorithm so that when a new image comes, the algorithm is going to be able to make a suggestion as to whether the image corresponds to a tumor or to a healthy tissue. So in my opinion, this is a case of supervised machine learning. Recognizing communities in social networks by affinity of users, for instance, is a case of unsupervised machine learning. A self-driving car would be a case of reinforcement learning. Predicting the price of houses by neighborhood, that would be a case of supervised learning. And a computer playing chess or playing Go, for that matter, would be a case of reinforcement learning. Right, so let's focus on supervised tasks, which in my opinion are the easiest to understand, and let's focus, I hope you will bear with me, in drug discovery, which is my field of expertise. If you've been in a laboratory, you might recognize this setup. This is a 96 well plate, and in each plate there is bacteria, in this case E. coli, growing. The darker the well, the higher the growth of the bacteria. The lighter the well, the less the growth of the bacteria, that is, the bacteria are dying. In each row, here we are dosing a particular drug candidate. So a drug that is dosed and remains dark, this means that this molecule is not killing the bacteria, so we label it as inactive. A molecule that inhibits the growth of the bacteria, it's potentially a good antimicrobial candidate, and we label it as active, right? Whites will be actives labeled as actives, purples will be eventually labeled as inactive because they don't inhibit the growth of the bacteria. So the first step here will be to try and convert this data that we have collected in the laboratory. It's probably in an Excel sheet, and we want to convert this data into something that is amenable for machine learning. And this is the key step of machine learning, in my opinion. This is what we call featurization. How does it work? If you are a chemist, you will recognize this is a small molecule that has some interesting features. For instance, one of the features of this small molecule is the number of rings. This molecule has 3 rings. Another potentially interesting feature of this molecule is the solubility. This is something that we can quantify, for instance, by counting the number of nitrogens and oxygens that the molecule has. The more nitrogens and oxygens, the more soluble in water probably this molecule is going to be. This is something that we can quantify. The number of fluors we can also quantify, and the molecular weight, for instance, right? The more atoms the molecule has, the heavier it's going to be. So now that we have predefined these features, that we have featurized our input, we can explore how these features relate to the outcome that we are interested in. For instance, we can do these 2D plots. In this case, I'm plotting here the number of rings against the solubility, and you can see that the number of rings is really not discriminative, right? But solubility is. So the higher the solubility, the more actives we find. The lower the solubility, the more inactives we find in blue. Actives are red, inactives are blue. If we go to number of fluors and molecular weight, we can see that here the discriminative feature is molecular weight. So the heavier the molecules, the more actives we find. The lighter the molecules, the more inactives we find. So if we combine feature 2, which is solubility, with feature 4, which is molecular weight, we can see that more soluble molecules that are heavier tend to be active, less soluble molecules that are lighter tend to be inactive. So now all what we have to do is to encode this in a format that the computer can understand. That is, we have to convert this molecule into a vector, and in this vector, in each position of the vector, we will have the quantification of each of these features: feature 1, feature 2, feature 3, feature 4. I was showing 4 now, but actually in practice we computational chemists, we can count thousands or even dozens of thousands of features, right? So the key step in featurization is converting whatever input we have into a format that the computer can understand, into a numerical vector, essentially. This can be done, of course, not only for molecules, but for any kind of input that we might think of. Can be images, can be text. Here I'm converting this sentence into a format that the computer understands, in this case a binary format. Okay? Don't panic, I know this sounds abstract, I know this sounds difficult, but we will really dig into it in upcoming sessions. We will really explore how can we featurize for record linkage in upcoming sessions. Let me just talk a little bit more about chemistry. So remember, when we have a molecule, we are able to featurize it. We can explore the structure and put it into a vector of zeros and ones in this case, that basically encodes the structural information that we have for the molecule. So what's key here is that this vector, that is the input for machine learning, essentially represents numerically my input. Okay? Of course, here I'm just encoding the structure, but if things can get more complex, for instance, I can get all of the information that I can gather from the scientific literature related to this particular molecule. For instance, the toxicity the molecule causes, the targets and the off-targets, etc., etc., etc. Because we know how to featurize not only chemical structure but also text, we can always embed and encode as vectors all of this information into something that can be the input of machine learning. Long story short, the more complex the featurization, the better eventually will be the predictive power of the models that we will build. Okay? In this case, we are building a very, very complex and rich vector for this particular molecule. Therefore, we have good likelihood of having good predictions eventually. Now, after we have featurized our input, in this case our molecules, we have to identify the best machine learning tool to make the predictions. And I have to say that most of the focus in most of the tutorials that you will find out there are focused on this particular step that is fitting. In my opinion, featurization, the previous step, is equally important or even more important at building competent and prominent machine learning tools. Okay, but because fitting is so rich in techniques and so on and so forth, it gets most of the attention. Essentially, what we are doing when we are fitting a model is trying to identify a mathematical function that is going to be able to discriminate between, in this case, actives and inactives, right? Fitting a model is finding this line here, so that when we come with new samples for which we don't know the outcome, we will be able to make a guess whether those samples will be active, the upper right region of the plot, or inactive, the lower left region of the plot. Okay. Of course, finding exactly the precise position of this discriminative function is the key to machine learning, and this is why so many algorithms exist, because finding this discriminative function can be very difficult sometimes, right? The better and the simpler this discriminative function is, the more generalizable it's going to be. This is something, if you go in the tutorials, that is usually referred to as overfitting, right? And I think it's better understood in this example. Let's assume that we have this more difficult case where actives and inactives are sort of more merged within this 2D plot. We can always come up with a very, very complicated decision function that is going to separate very well our known data, right? But when we come with new data, maybe it's not going to do so well, right? Because we have overfitted. We've trained a model that is too complex for the small data that we have. It's always better to come up with something that is simpler, like this, so that when we come with new data, we will generalize better. We will have less overfitting. Okay? This is key to machine learning, and this is why so many algorithms exist, because they all try to tackle this problem, this balance between complexity and generalization of the problem. So fitting has two steps. First, we have to choose the algorithm. We have to choose the decision function, the mathematical function that is going to be able to discriminate between, in this case, actives and inactives. And I would say that when we talk about classification, the major families of algorithms are support vector machines or classifiers, naive-based classifiers, and random forest classifiers. If you master those three, I think that you are good to go in most of the tasks. Then once you have chosen your algorithm, you have to tune or identify the parameters, the hyperparameters of your model, so that it performs well in your particular task. Finding optimal parameters today, thanks to good libraries that are available out there, can be automatized, although it can be computationally intensive sometimes. Once we have identified our features of interest and our algorithm of interest, and we have fitted our parameters, it's very important that we validate our model, that we make sure that the model is going to work well with new data. And usually the trick that we use in machine learning is to split our data in a training test and a test in a training set and in a test set. Training set usually is bigger, typically 80% of the data, and a test set is smaller, typically 20% of the data. What happens here is that you choose your features and you train your model on the training set, you feed your model on the training set, and then once you are done, you evaluate it on the test set. Okay? Actually, if you have enough data, it's better to do 3 splits. 80% of the data you can keep it in the training set, 10% of the data you call it validation set, and another 10% of the data you call it test set. And then you do multiple cycles until you find the best model on the training set and validation set. Yeah, and you will have an idea of the accuracy by doing these cycles that will help you identify the best algorithm and the best parameters. And once you are done, you evaluate everything on the test set that you have separated from the beginning. Okay, this you can do it if you have enough data. So let me go back to my favorite supervised drug discovery task. So to summarize, we have from our laboratory experiments, we have different molecules that we know are active, or we have labeled as active or inactive. We can convert these molecules, we can feature these molecules into a numerical vector, each of them, that the computer can understand, and on top of that we can fit a model, typically in this case a classifier, for instance a random forest classifier, as I presented before. And once we have fitted and validated this model, we can essentially apply it to new data. So when I come with a new molecule, it is not necessary anymore that I do the experiment in the laboratory. I can just featurize this molecule, get the vector correspondingly, and then make a prediction using the fitted model, right? In this case, I'm predicting that this molecule is going to be active against E. coli because it's going to inhibit its growth. So the machine learning pipeline can be seen with this kind of representation. At the core of the pipeline, we have the raw data, the collection of data that we have to process and featurize. Then once we have processed and featurized the data, we have to train a model, test and validate it, and finally, ideally externally validate it. That is, give the model to someone else so that they can test it in external data. Okay. Once this process is done, we always have to deploy the model. This means making the model ready for production, implementing it in our software of interest, right? This is called deployment, and as I said before, we are going to touch this particular topic in the advanced course. At the end of the day, once we have deployed the model, the user, our collaborator, will be exposed to a software where the input is going to be, in my case, a drug, and the output is going to be a target. For instance, the inhibition of E. coli growth in this case. Okay, so we've done a lot of work as data scientists, but what we exposed to users is something that is really very simple. I think that this is the end goal of machine learning, and in particular the end goal of machine learning applied to record linkage. If you are a software developer or if you are a computer scientist, you might prefer this kind of representation where we have a continuous delivery and automation pipeline. Basically, in the first circle, we are doing what I've just explained, collecting data, engineering features, training and tuning. But then at some point, we are entering the development and the operational circles where we are basically testing this model in production mode. Okay. So I hope that this sounds interesting and this sounds feasible, and I hope that at least qualitatively I was able to present like the very basic concepts of machine learning. Now the question is, if you want to go hands-on, where do you start? And I have to say that if you go to the internet, your experience will be really overwhelming, because there's many frameworks that are very good for machine learning. There's many technologies, there's many solutions, so it can get a little bit overwhelming at the beginning. I really encourage you to follow the Moodle suggestions that we are making in this record linkage course. Also, if you are an epidemiologist or a clinician or a biomedical researcher and you go to the scientific literature, you will find many very good papers that are at the interface of experimental data and also machine learning, and those papers are pretty advanced and they can be overwhelming at first. Same happens if you try to follow courses. You will find a lot of hierarchies, a lot of organization. There's a lot of algorithms. There's a lot of taxonomy. There's a lot of possibilities, and at first sometimes it can be a little bit overwhelming, as I said. So there is this universe of possibilities. There is this universe of data. There is this universe of information Here we will really try to keep it down to earth. We will just focus on the particular topic of featurization. We will focus on classification, which is a kind of supervised machine learning, and we will focus on clustering, which is a kind of unsupervised machine learning, and we will focus a little bit on validation. Nothing more really. So we will really keep it simple. We will really keep it down to earth. So what do you need? You might ask, well, you don't need much. It's not that you need big computers. If you have a laptop and a decent internet connection, you are good to go. Back in the day, I used to use computers like this one. This is the Barcelona Supercomputing Center. This is a huge supercomputer, and what's beautiful about it is that it's inside the church. You don't need anything like that anymore. Machine learning can be done from a laptop computer. Algorithms are super optimized and ready to use with any hardware, really. So don't worry in this regard. Take-home messages. I hope I was able to convince you that you don't have to be an expert to start playing and tinkering with machine learning. If you have a laptop and very basic coding skills, I think you are good to get started. Machine learning is really tightly connected to statistics, to classical statistics, so we will not see any fancy algorithms. Everything is going to be really easy to understand, hopefully. A typical machine learning pipeline involves first and most importantly, in my opinion, featurization, then training and test splitting, choosing the right algorithm and fitting the parameters accordingly, evaluating performance, and finally deploying so that the machine learning model doesn't only stay in your computer but is used by others. What's next with regards to this course? So we are not yet done with Module 5. We still have to do a forum discussion that is going to be followed up by a Q&A Zoom session, a live Q&A Zoom session where I will get to know you guys and you will get to know us. And there will be a Moodle questionnaire that we will appreciate that you answer. You will find extra materials in the Moodle page corresponding to Module 5. And in the next module, Module 6, the last one of the basic record linkage course, we will really go into we will revisit the record linkage pipeline, and we will ask the question, how can machine learning can help improve this pipeline, making it more efficient and hopefully better in terms of accuracy of results. That's all for today. I really hope you enjoyed this first session introductory session on machine learning. See you next time.